In this video, I'm going to talk about osmosis, and we're going to think about the different effects that osmosis has on cells. So let's remember that osmosis, what it is the diffusion of water across the cell membrane. So basically, we're just thinking about water moving from the side of the cell where there's a greater concentration of water to the side where there's a lower concentration of water. And so overall, cells generally want to have an equal amount of water moving in and out. That allows for the cell to maintain its size. If there's a net movement of water out of the cell is going to shrink. If there's a net movement of water into the cell is going to swell and could potentially burst. So to have equilibrium, an equal amount moving in and out, cells need to be surrounded by an isotonic environment. So that is the first of three different situations we're going to think about as far as conditions that a cell could be present in. So you generally want to think about cells as being surrounded by watery fluid. But they're not going to be surrounded by pure water. They're going to be surrounded by water that has stuff dissolved in it. And here, zoom in on this picture here, we have a visual for what it's like when a cell is surrounded by a solution that is isotonic. So let's add in a little key here. The blue represents water molecules, and then the red represents solute, which is just anything dissolved in the water. That could be salt, sugars, ions, anything dissolved in the water. So here the yellowish orange color represents the cell membrane. Okay. So in this first cell here, we have inside the cell where the cytoplasm is, it looks similar to the outside as far as how concentrated or how dense the little water molecules are and how much solute is dissolved in it. So I would say that the amount of solute dissolved in the cytoplasm matches or is the same as the amount of solute, the red stuff, dissolved in the fluid outside the cells. And because of that, there's going to be an equal amount of water that's moving in and moving out. Water moving in and water moving out. There's not going to be a net movement from one side to the other because there's not really a difference in concentration of the water outside and inside. So if the fluid around the cell has the same solute concentration, then you're going to have an equal amount of water moving in and out. And as a result, there's going to be no change in the size of the cell. Okay. Now, the next type of environment that a cell could be surrounded by is a hypertonic solution. No, sorry. No, I'm sorry. We next have hypotonic. My bad. Switched the order. Okay, hypo. Hypo means low or less. So here, the solution around the cell is going to have less solute in it. So let's look here. This fluid surrounding the cell, you can see there's hardly any of the red solute in it. And so then now let's think about osmosis. What's going to happen with the movement of water? There's a higher concentration of water out here compared to inside. So overall, there's going to be more water moving in. Even if there's some going out, overall, there's more going in, and our cell is going to get bigger. Okay, so when there's a hypotonic solution with very little or just less solute than the cytoplasm, there's a net movement of water into the cell. And our cell swells or potentially even bursts if there's no cell wall around it and enough water enters, it can burst. That's also known as lysis, when a cell bursts. All right, now the other possibility is, what if instead of having less solute, the material or solution around the cell has more solute dissolved in it than the cytoplasm? That would be a hypertonic solution. So here there's more solute in the fluid around the cell. So hyper means extra or excess. So when it's hypertension, they have high blood pressure. Here there's a higher solute concentration out of our cell. So you can see on the outside of the cell, there's a lot of solute, less water. So the water is going to move from where there's more water to where there's less water. So overall, there's more water leaving. Could be some passing in the direction inside, but there's a net movement out, and we can see our cell shrinks. Net movement of water out of the cell. And the cell shrinks or decreases in size. Okay, so in general, like in your body, your red blood cells that are floating around in your bloodstream, they're surrounded by something called plasma. That's a watery fluid, and it is isotonic to the cytoplasm inside of your red blood cells. And so because of that, you have, this is a picture of a red blood cell here, you have equal water moving in and out of your red blood cells at all times. And at the top here, we have, a visual for kind of what happens with animal cells. And then here we have a visual for what would happen with plant cells. Plant cells, when they're in an isotonic environment, they also have equal amounts of water moving in and moving out. And so they would stay the same size. Now, in the hypotonic environment, remember that's where there's very little solute in the fluid around the cells. So like imagine red blood cells were put into pure water and there's no solute at all out there. Well, then we're going to have water entering into our cells, and in an animal cell they could burst. Now a plant cell won't burst because of having their cell wall. Okay, so that's one thing that they're not at risk of. And then in both cases, so here the water was entering, but the plant cell has the cell wall to prevent the bursting, whereas human cells, they can burst if they're around too much water. This is what can happen if someone were to overhydrate, if they drank a ton of water and they were surrounding their cells by fluid that had a lot of water in it with very little solute. Let's say someone is doing a marathon and or like even an ultra marathon, and they're just drinking water and they're not eating anything or having any Gatorade. There have been stories where people have actually overhydrated and even died from it. Now, on the other side of things, what if someone is not drinking enough water and their cells end up surrounded by fluid that has a lot of solute but not very much water in it? Here, the cells would shrink, and this can cause dehydration with things like muscle cramping and headaches. In a plant cell, water would leave, and this would cause, like drooping or wilting to happen. Okay, and you can see here how the cell membrane is pulling away from the cell wall as that central vacuole shrinks. So that's an overview of how osmosis affects animal and plant cells. Next, I'm going to talk about, we're going to learn about active transport. So active transport is when a cell has to use energy to move material across the cell membrane. And the specific kind of energy that cells use is called ATP energy. So this is really like the first time we're talking about ATP. So ATP is the cell energy form that is used anytime work needs to be done in a cell. And this is ATP. And a lot of times this symbol, it looks kind of like a firecracker dynamite symbol, is used to represent ATP. So ATP is this molecule we'll talk more about later on, but there's a ton of energy stored right here where this lightning bolt is between these two P's. There's a ton of energy stored right there. And anytime a cell needs to do work, it can just pop off that last phosphate, that last P, and that energy is released. And it gives just a punch, quick pack of energy to be released for whatever work needs to be done in the cell. So with active transport, we're going to have to use some of that ATP energy. So the first situation where a cell would have to use energy is if they want to move something up or against the concentration gradient. Like in this picture here, we can see if you're trying to move this green thing into this cell, you're trying to move it from where there's a low concentration to where there's a high concentration. This is the opposite direction of what would happen by diffusion. So in order to achieve that, you have to use a protein pump. So think about if you are like pumping up a football or a basketball, you have to actively put energy into pumping that air in if it's a manual pump. Here, this protein is going to need to be powered to, a lot of times they'll spin as they move something in. And so it looks a lot like the proteins that are used for facilitated diffusion, but you can tell the difference because you see things that are going against their concentration gradient, and you'll often see that ATP symbol shown. Another reason why energy would need to be used is if you're just trying to get a large quantity across the cell membrane. And the term that's used for this is called bulk transport. When you buy things in bulk, generally you do that going to a store like Costco or Sam's. You buy a lot at once. Here, the cell wants to get either something really large, like, in this picture down here, you can see this white blood cell is kind of eating a bacterium. So it wants to take in that big cell all at once. Or say over here, you can see a bunch of little things. It wants to take all of those little things in at one time. Either way, if you are taking in a large quantity at one time, that's called endocytosis. Endo means within, and cytosis means cell process. So here you can see the cell membrane kind of wraps around whatever it's taking in. And that part that wraps around pinches off and forms a vesicle. So this is the vesicle. That's the little transport capsule surrounded by a lipid bilayer that is going to then be able to move that material inside of the cell. And like at the white blood cell, it would engulf the bacterium or virus or whatever other harmful thing is being taken in, and then it would use its lysosomes to break it down. Other times, cells want to ship things out. And if they're shipping something out that's really big or a bunch of product all at once, then exocytosis can be used. So here we have things exiting. So exo, think exit, whereas with endo, you want to think enter. So here we have a large quantity exiting the cell at one time. And it's kind of the reverse. If you were to rewind endocytosis, it would look a lot like exocytosis. So here the vesicle travels to the cell membrane, fuses with the cell membrane, and then releases, the material gets released outside of the cell. An example of this happening in your body all the time is when one nerve cell sends a signal to another nerve cell. So if this nerve cell is going to send a signal to the next one, let's say a signal in your brain wants to communicate with your spinal cord, what happens is at the end of the brain cell, there's these little vesicles that each contain chemical messengers called neurotransmitters, and they have all sorts of names like dopamine and serotonin and endorphins. And so basically the vesicle can fuse with the brain cell's cell membrane, and then the little neurotransmitter messenger chemicals can be released, and they can attach to receptor proteins on the cell membrane of the spinal cord cell, and that can trigger a message to be sent in that receiving cell. So as far as cell transport goes, just think about here. Remember, it's bulk transport. We have a lot of material being moved out of the cell all at once, and since the material is moving out, we call it exocytosis, and ATP energy would need to be used to do that. All right, so all three of these, protein pump, and then our two types of bulk transport, would require ATP energy, meaning the cell is having to spend energy to get this work done, transporting things across the cell membrane.